Hello, this is Greg Risterbin from Olympus. I'd like to welcome everyone today to this webinar titled Plant Imaging and Clearing for Light Sheet Microscopy. In recent years, light sheet microscopy has become a key imaging method for plant biology. With new clearing methods available, it's even easier than before to study plant development. However, more recent research studies are looking to apply light sheet microscopy techniques to focus on root imaging, requiring new methods for sample mounting and imaging to be developed. To begin, Dr. Tim Stepp of Logos BIOSYSTEMS will discuss considerations for clearing plant samples using the X-Clarity tissue clearing system for in vitro imaging. Next, Mathieu Cortes will discuss how using the alpha-3 light sheet microscope can extend the scope of plant imaging. Dr. Tim Stepp is an application specialist with Logos BIOSYSTEMS with a PhD from UNCCH in Marine Sciences and 15 years of industry experience. Mathieu Cortes has his master's degree in cell and molecular biology and is currently a PhD candidate at the Laboratory of Plant Reproduction and Development in the Mechanotransduction and Development at École Normale Supérieure de Lyon. This informational webinar has been scheduled for approximately 1 hour. If you have questions, please type them into the Q&A panel in the lower right corner of the presentation screen. If we do not get to your questions during the live webinar, they will be addressed personally either by email or phone after the event. Now, without further ado, I'd like to turn the presentation over to Dr. Tim Stepp. Tim, you may take it away.
Thank you, Greg. We greatly appreciate the opportunity that Olympus is providing us to present to you today. And so what we're going to be going over is doing tissue clearing, and most specifically incorporating the X-Clarity tissue clearing system into your plant clearing process. And so first, what we'll do is we'll establish a simple functional definition of what is tissue clearing and go into what is involved in that. And then we'll get into the challenge of clearing plant tissues. And just to give you a little heads up there, it's not necessarily the clearing. Plants in many ways plant tissues are simpler to clear than, say, mammalian tissues. But when it comes to the labeling, if you're doing antibody labeling, that is a different story. So we will address that a little bit. And then we will get into different approaches for clearing plant tissue. So what's been used, there's been several different approaches to clearing plant tissues. And then we will talk a little bit about the X-Clarity system, whether or not it can be used for clearing plants. Obviously it can, but then we will get more in specifically about what the X-Clarity system is and the workflow that is involved in clearing plant tissues with the X-Clarity system. And then we will address the antibody labeling of plant tissues and the considerations for that. Give you some examples of what cleared plant tissue may look like. And then finally, some considerations for you as you're setting up your tissue clearing workflow of what you may want to consider when you head down this path. So what is tissue clearing? And so from a simple functional definition is tissue clearing creates an intact, optically transparent tissue sample that can be imaged at depths over a few hundred microns to several millimeters. Now, the emphasis here is on the imaging component. So what you're doing the tissue clearing for is so that you can get clear and insightful images, not necessarily so that you can get clear tissues. It's what the clear tissues allow you to do for the imaging is why you want to go about this. And so there's a couple of different things that happens when you clear tissues. It's what I call remove and replace. So the first step for most tissue clearing methods is simply removing the major light scattering discontinuity source, and this is lipids, and replacing that with an extracellular water with a refractive index matching solution that matches the refractive index of the objective. So again, one of your most important considerations here will be what tissue or what imaging system you will be using and what sort of parameters are around that imaging system, and most importantly, what the refractive index of your objective will be. So as we addressed, as I mentioned a little bit earlier, is that plant tissues are actually a little bit more simple to clear than mammalian tissues. But there is the one difference within plants versus animal cells that does hinder the labeling, and which we'll address more at the end. And that is simply the cell wall. And so while plants may clear out very well, it's the labeling and getting your antibodies, if that's what you will be doing, into the cell. Now, looking at the literature, there has been several different ways that people have gone about doing tissue clearing for plants. One of the most popular is using urea-based solutions. More historically, this has been done with an acidified chloral hydrate. At this point, in most countries, this is a pretty toxic and regulated substance, so it might not be as easy to use. There's also papers out there that have and groups that do plant tissue clearing with water-soluble alcohols like TBE or TDE, excuse me, and then also some that use organic solvents such as methyl benzoate. And certainly there have been a couple of papers out there with using the clarity-based methods, and obviously the X-Clarity system is an extension of the clarity-based methods. So the original clarity-based method that was used for plant clearing was the P-Clarity, which was released back in 2015. Now, this is what we call a passive clearing method. So essentially what they did is they established the typical hydrogel infusion and then polymerization, which you do in the clarity process to retain your macrostructure and your cellular integrity. And then they just simply soaked that in SDS sample or SDS buffer and slightly elevated temperatures. And so that's what we call passive clearing. So in other words, it's just sitting in the solution. Every once in a while, you change out the solution. Now, the original clarity paper that was released back in 2013 from the Diceroth lab was actually an active clearing. So in other words, after they did the sample fixation and the hydrogel monomer infusion and then the polymerization, then they put their sample in a container and ran a current through that at a slightly elevated temperature. And so what that current does is it actually speeds up the removal of the lipids. Now, certainly there was plenty of people that tried this passive clarity method and ran into all sorts, or excuse me, the active clarity method, and ran into some issues in trying to develop their own system. And so that's the point at which we decided to develop the X-Clarity system. And so the larger question for what we're discussing today is, so instead of using, say, neuronal tissue or heart tissue or anything like that, can we clear plant tissues like leaves, roots, things like that? And the answer to that is yes. We can do that with the X-Clarity system. So for example, what we see here is just a typical mouse brain, and the 3-panel images shows that it was, what it looks like fixed or non-cleared. After you go through the clearing process, it is more a translucent sort of material. And then when you do the refractive index matching, so that's when the final transparency actually happens. And so that's when you get these really, really clear samples. And so same thing can be done with the plant tissue. And here's an example of an Arabidopsis leaf that was cleared by our group, shows what it looks like after it was fixed and then what happened after it was cleared. And so this was done in 10 hours at certain current and temperature. So at this point, we know that there of, what other types of tissue besides leaves, and Arabidopsis have been cleared. So certainly there's been melon, pea, wheat, Arabidopsis, medicago, tomato, tobacco, bean, and ficus. So a full range of different types of plant tissues and species. Now, this is the X-Clarity system itself, and it is composed of several different components. Now, the main clearing system is what we have right here. over that I'm circling the cursor on right now. So over here is the control tower, and the control tower controls the current, the temperature, and the pump speed. In the back here, which may not be so visible because it's clear, is where we hold the SDS buffer. And then hopefully you can see these tubes that run from the control tower in through the sample chamber right here. And this is the sample chamber where you place your sample, and it also has the electrodes through which the current crosses. And so when essentially you set this up to run, the buffer will circulate from the reservoir through the system. It's heated, and it goes into the clearing chamber and comes back out and back into the reservoir. Over here on the left-hand side is the polymerization system. Now, what this is, it's a pretty sophisticated device that does a very simple thing. So as I said, when you are doing the clarity-based methods, you need to infuse your samples with hydrogel, or it's acrylamide is what it is. And so after these samples are infused with acrylamide, you need to polymerize that so it is set up and it retains the structure of the cell. And so you can do that with a vacuum chamber and flushing out nitrogen, or you can do that with a simple system where you put in your samples in here, you close the lid, and you press run. And what it does is it removes the oxygen, puts it up at the necessary temperature, and 3 hours later, the samples are polymerized. So it makes life a lot easier. The other parts of this are the different types of sample containers. So we have very variable and configurations for our sample holders. So for example, we have a 6 sample holder, we have 36 sample holders, we have certainly larger sample holders if that's what you want to do also. The other part of the system are the reagents. And so we have the hydrogel kit, the clearing buffer, which essentially is SDS. Then we have also what's known as the deep label antibody kit. This will help speed up and antibody labeling and make things more specific. And then we also have our version of refractive index matching solution, what we call the mounting solution. Now, how this may proceed with the XClarity, the workflow is simply you're going to cut your sample and wash that with PBS. The next comes the sample fixation. Now, with animal tissues, this fixation may go for about 24 hours. With plants, this can proceed from anywhere from 3 to 10 hours, just depends on how thick it is. So for example, if you have 1 millimeter size leaves, then about 3 hours is going to be good enough. The next comes the hydrogel infusion, and then after that is the polymerization. And then the tissue clearing itself is going to proceed from anywhere from about 2 to 72 hours, depending on the thickness of your sample. Most samples and most plants will clear out in between 2 and 20 hours. So 72 hours would be very, very far out there. So the next and often overlooked, but very, very important step is to do the post-clearing wash. So this removes out all the SDS from the sample so that you can do the antibody labeling and imaging downstream without too much difficulty. Now, as we mentioned earlier, the big thing is the labeling. And so this is going to take anywhere from 2 to 14 days, depending. And the other thing that you may not be as used to with the labeling when you're doing very thick tissues is that the antibody concentrations need to be a lot higher than what you may normally use. For example, the primary antibody from a 1 milligram per mil stock solution, you may need to dilute down to only 1 to 100 rather than say 1 to 500 or 1 to 1,000 that you may normally be used to. After labeling comes the refractive index matching, and then certainly the imaging and then the analysis. Now, as I mentioned, it is the labeling, if you have samples that you need to do antibody labeling with, that can be the biggest challenge with plant clearing. Now, from the P-Clarity and others, and for those of you that have worked with plant tissues, you know that there's a workaround with this, and that is simply you want to do an enzymatic degradation of the cell wall. So basically, you're permeabilizing that cell wall so that the antibodies can get into. And from this, you can do the enzyme cocktail that includes cellulase, xylanase. I don't even want to try and pronounce that next one, pectate lyase, and then alpha amylase. So there's all sorts of different cocktail mixes that you can use. And this may be something that for your specific samples that you may need to try by trial and error to figure out what works best for you. Now, with regards to comparing what, the X Clarity brings, or, what does the X Clarity do for you? And it is simply a matter of time. And so it will greatly speed up your clearing process. So for example, what we have here are some ficus leaves. On the left-hand side, we see that this has been actively cleared. So in other words, we have run the current and allowed through the chamber. And this was done at 15 hours for 37 degrees at 1.2 amps and 100 RPM. Okay, and so you can actually see how much clearer this is than, say, doing a passive clearing, where you're just letting the sample soak for 15 hours at 37 degrees. And just to give you a better idea of how that may look as it goes through that whole depth of the leaf, is you see these GIFs. On the left is the active clearing, and then over here on the right is the passive clearing. And so you can see you have great clarity, no pun intended, over here on the left, whereas it is particularly, it's very dense, and light doesn't penetrate as well in the passively cleared sample. Now, there are some considerations that we highly recommend that people take into consideration before they even start down on this journey on trying to do in-depth imaging and clearing of samples. One is understand what imaging system is available for you to use. The second is you want to make sure that your clearing method will be compatible with your labeling method. As I mentioned earlier before, the clarity method and a lot of other clearing methods, they will remove lipids. Now, if lipids are something that you want to target, obviously you will need to think of a clearing method, and there are some out there that don't necessarily remove the lipids. Now, the drawback on that is you won't be able to get the depth of imaging that you might, but you can certainly work in smaller sections for that. So essentially, it's compatibility of your clearing method with what it is you're actually trying to visualize. And the other is the compatibility of your clearing method with your labeling method. So for example, let's say that you start with endogenously or virally labeled samples. You want to make sure that your clearing method is going to be compatible with maintaining and retaining that fluorescent signal. Ultimately, though, what we would definitely recommend is to figure out what it is you want to image, what you have to do the imaging with, and how you're going to go about your labeling. And then once all that's established, is then compare and contrast several different methods. So in other words, compare a passive clarity or an active clarity versus a urea-based or a organic solvent-based or an alcohol-based clearing method. And ultimately what matters is what works best for you. So try several to figure out what works best for you. And so with that, what I'm going to do is, at this point, I am going to pass the presentation on to Mathieu Cortes, who will be talking about doing plant imaging with the Alpha 3 light sheet imaging system.
Thanks, Tim, for your great talk about clearing for plants. It's really interesting. So now I will go to the plant imaging and the light sheet microscopy. So I will focus on aerial organs of plants. I will start by an introduction about plant imaging and imaging in general. After, I will explain you what you can do with light sheets, how you can mount some sample, how you can image aerial organs in the alpha 3 light sheets. And after, I will show you some application and results I had during my PhD with the alpha-3 microscope. So first, plant imaging and imaging in general. So in plant, we use a lot imaging for many studies. It can be cell division plan, expression pattern, growth analysis, microtubules rotation, or calcium flux, for example, many applications for the imaging. And so if you do fluorescence microscopy, what you want basically is to follow fluorescence in 4 dimensions. So you want a 3D image during time. It can be many kind of signal, but you want a signal during time and in 3D. And so in order to have this kind of image, we have some needs in imaging. And so here I summarize the needs we have in this triangle of needs. And so we have the spatial resolution. So everyone wants the best spatial resolution possible, and it's really useful for us when you have a great spatial resolution. There are the temporal resolution. So the temporal resolution can be really important when you do live imaging and you want to follow some signals during time. And so basically the temporal resolution, it's the time of acquisition, how many time your microscope take to make one image. And there are the depth of imaging. So here basically is the number of slices you need to do your imaging. And so sadly for us, we don't have one microscope which allow to have the 3 needs at the same level. So we need to do choice when we do experiments in order to have the best parameters we want for our experiments. And so maybe what you need is spatial resolution and depth for imaging. So basically here, what you need is 3D imaging. And so you will use the confocal microscopy. It's the most common microscopy used in plant imaging. And so just briefly, the confocal imaging. In confocal microscope, we illuminate the sample with a laser and we scan the sample pixel by pixel in order to produce one image, one slices. And we do that for every slices we want. It's interesting because we can choose the size of the pixels in order to have an optimal size for the lens we use. So if we have a really good lens with a really good numerical aperture, we can choose the optimized size of pixel for this objective. And so we can have a high amount of pixels. The disadvantage of this is if we have a big amount of pixels, we need to scan all these pixels. So it takes time. And during the imaging, we illuminate the sample during all the imaging and in all the depth of the sample. So in a confocal microscopy, there are some advantages. We can have a good X and Y resolution due to the fact we can choose an optimized size of pixels with our objective, and we have quite a high sensibility with the PMT and now some hair bright PMT. But there's some limitation. Due to the fact we scan the sample, we have a low temporal resolution, so it can take time to make images in the confocal. So if you already did imaging, confocal imaging, you know, if you want a big stack with a big amount of pixels and many slices, it can take several minutes, tens of minutes, and maybe hours. And so during all this acquisition, you illuminate the sample, and so you induce a lot on phototoxicity and photobleaching. It can be complicated when you want to image live organs, you want to follow developments, or if you work on weak signals. And so here just some example of what we do with confocal imaging, really classically in labs. So here what you see, it's 2 dissected apex. So in the left, sorry, you have in the red, in the middle, you have the sutapical meristem and several floral meristem. and you see in red labeling for the membrane, and in green yellow you see the reporter for the agamous gene. In the right, the same thing. So in red you have the labeling for the membrane, and in yellow green you have the labeling for the Wuchel gene. So what we do in confocal microscopy is we can do time lapse, so we can follow growth during time. So here, for example, we can follow the cell division, but as I said, we can't have a really high temporal resolution. So maybe it's what you want. You need temporal resolution and depth of imaging. So basically what you need is 3D during time, and you want to have the shorter time step as possible for your time lapse. And so when you need that, you can use the spinning disk. But today I will present you the light sheet fluorescence microscopy. I will show you how you can do 3D images to have a high amount of slices in your stack and have a high temporal resolution. So basically, what is light sheet microscopy? Donc, light sheet microscopy is really different than confocal microscopy, but the concept is not really difficult to understand. So if you are in green, your sample, so it's a side view, you will create with 1 or 2 lenses, you will create one light sheet here in blue, and so you will illuminate your sample just in the thickness of the light sheet, and you will capture the photon by a lens and a camera. And so here you already see as the sample is just illuminate in the thickness of the light sheet. So the sample is not illuminating all its depth during all the acquisition time. And so if you want to do 3D imaging with the light sheet, you just need to move your sample in the light sheet, and like that you can create slices and so stack and so 3D imaging. The big difference is the illumination and the detection. The detection is the wide field detection, and so in contrast to confocal with the PMT, where you need to scan the sample pixel by pixels, here we can image the sample slices by slices. So it's really faster than the confocal. And so we induce less phototoxicity and less photobleaching due to the fact the sample is just illuminated in the thickness of the light sheet, and due to the fact the acquisition is really faster. So if I do a brief summary, light sheet versus confocal microscopy, we have roughly the same axial resolution than in the confocal. You have the same X and Y resolution than a wide field microscope, but depending on the parameter you choose on your confocal, several times you have the same X and Y resolution in the confocal and in the light sheet. You can go deeper in the tissue, so I think I will show you. The acquisition speed is really faster, and the photo bleaching is really low. It can be really interesting when we want on development, for example, or with weak signals. So now I will present you the alpha-3 microscope and how we can mount our sample in this microscope and what kind of imaging we can do. So the alpha-3 light sheet microscope, it's an upright light sheet microscope. So it's really interesting. The architecture is really nice for aerial plants organs. Due to the fact we are in the same organization and the architecture than an upright confocal, so we can mount our sample in the microscope and image it by the top. So it's really interesting. I will show you after. So of course we can do multi-channel imaging with this microscope. We have 2 light sheet illuminators, which can be interesting in plant imaging, because if you already did plant imaging, you you know it's the plants or organs can be really you have really high scattering, so it can be interesting to have these 2 light sheets. You can choose many kind of lens for the observation. So two brief technology about this light sheet, which is interesting for the imaging. First, the high ANA for the illumination, which allow to have a really thin light sheet, about 2 micrometers. It's really interesting because we can do imaging from whole organ to subcellular components with this kind of light sheet. So it's really interesting. And in the Alpha 3, you have some technology modules in order to remove the shadow effects and the stripe in the sample, and it's worked very well. So if I come back on the architecture of the Alpha 3, as I said, it's the upright microscope. And so if I show you a close-up of the mounting modules, you can image your sample with a long working distance lens, as you do in other microscope, like in confocal. So it can be quite easy to transpose our protocol in confocal microscopy in this light sheet microscope. So if we want to image dissected apex, for example, IUC on this sketch, you see it will be not really difficult to do the protocol in this light sheet. So now I will show you some mounting of samples. So I will start just by the cotyledons. It's the organ I use in my PhD. So here the sketch of the mounting. So as you can see, we have in gray one holder. This holder can be filled by medium. It can be MS medium. It can be ACM medium. And you just need to put your sample on the medium and fix it with a drop of agarose low melting, and after you can put the holder in water in order to image your sample. So something you can see here, it's you don't need to embed your sample in order to image it. So it can be really, really interesting for some experiments. And the second thing is you can put several organs at the same time in the light sheet. So it can be really useful when you do a lot of imaging and you have a high imaging routine in your team, for example. So I try many mounting for many kind of sample, and so you can put in the microscope cotyledons, hypocotyls, sepals in flowers or dissected sepals and dissected apex. And so if you just need to do some tests, you just need to do one-shot imaging, maybe just in order to see expression pattern, you just can use the classical holder of the light sheet of the Alpha 3, and you just need to cut one piece of medium, put it in the holder, put your meristem, for example, in the medium, and you can image in the observation chamber. So you can mount several organs like this, and so it's really interesting because you don't need to embed your sample, and you can image several samples at the same time. And it's the same parameter of imaging than the confocal, because you are on medium in water. So now some example of what you can show in the Alpha 3 light sheet. So here is the labeling for 45S LT6B GFP. So it's membrane labeling. And so in the left, you can see a tip of a sepal. In the right, you can see a floral meristem with the sepal here and the floral meristem in the middle. And so you can see the labeling from the membrane is really good. And so here, of course, you can image some expression pattern like Vuchel, for example, here. And we can image with, for example, 45S and BDGFP labeling. You can image the microtubules, but I will come back on the microtubules after. So now I will show you some application and some results I had with this microscope. So first, I will speak about some standard imaging. So the idea is to try to show you same kind of experiment we did on the confocal, but on this light sheet, and after some application with the specificity of this kind of microscopy. So first, you can image dissected apex, and you can image floral meristem and shoot apical meristem. No problem with that. And you can see the signal of membrane is quite clear. And something you can see, we show a lot of signal in the depth of the tissue, so it can be really cool for some experiments. So good news, we can image shoot apical meristem, floral meristem if we want. And so first experiment, standard experiment, I would say, with that, it's a gross analysis. So here it's a typical gross analysis imaging. So I dissect one apex and an image one flower bud every 12 hours during 84 hours. And so it's really easy to do that in the alpha-free light sheet. And what is really interesting is we don't need to embed our sample in agaroses. And so we can do the same in protocol than in confocal. For example, we can put the sample in the microscope and after put back our sample in the gross chamber in order to do the imaging during 84 hours. So it's really easy to do. After, I can show you here one of the kind of gross imaging. So here it's 1 hour, it's a time lapse of 1 hour during 12 hours. And so you can see, we can follow the growth of the sepal and we can follow the cell division. This kind of experiments is not really difficult to perform in the alpha-free. If you want to do imaging more than 12 hours, you need to put some light in the microscope in order to illuminate the flower, and you need to put some liquid medium in the chamber, and so maybe you need to have some macrofluidic system in order to remove the medium in order to don't have a contamination. And so the question can be, okay, we can do a lot of time lapse, but I have a lot of data. Can I quantify my images, quantify my data? And the answer is yes. Here it's just an example. So you can imagine a whole dissected apex and try to do some segmentation in something I tried during my PhD. So don't look too close on this segmentation. It's not a corrected segmentation. So there are some mistake, but I use the TMHTK Python library, which is really interesting with light sheet microscopy and 3D segmentation. It's with the depth we have in imaging, the signal we have deeper in the tissue, we can segment it a big amount of cells. Here it's a segmentation of more than 7,000 of cells. And so you can quantify your image, but just for example, I will just show you some difference between the confocal signal compared to the alpha-free light sheet microscope signal. So here, the same slice is the same slice, it's the same sample. And so, as you can see, the signal of the confocal is really noisy compared to the alpha-free. So my parameters for the confocal is not the optimized parameters because I want to have one kind of imaging I can compare to the alpha-free. So it's with a reasoning scanner here. And so what is really interesting too is if we go deep in the tissue. So here we are at 12 micrometer deep in the tissue with the confocal microscope with a reasoning scanner on. We lose the signal of the membrane. But with the alpha-free, we have the signal of the membrane, and so we can see the shape of these cells, and so we can do analysis of shape, for example, and follow the division pattern, for example. So one experiment I did is I want to compare if the quantification between the two modality is the same. So here it's one experiment of ablation. So I did one ablation and normally the microtubules labeling, the microtubules will reorient themselves around this ablation. And so I did this. I did same samples, and I did the imaging in the confocal and in the alpha 3. And so the response is we have the same result. We don't have some significant difference between these two modalities. So it's really interesting because we can really trust what give us the alpha 3 in kind of signal if we use a lot the confocal. So now I will speak about the application with the specificity of the lighting. In the first part of application, I show you you can do the same kind of experiment in the alpha 3 than in the confocal, and it can be really interesting if you work with weak samples or weak signal. Of course, if you have very weak signals, the light sheet can be a good option because there are a low photo bleaching, so it will be easier to image your signal. And if you have a really sensitive mutant, for example, it can be easier to use the light sheet because you have less phototoxicity, so it will be easier to image this kind of sample. But now I will show you some application in order to use the fact the light sheet can do a really fast imaging. And so I will show you what I did with a fast biological event. So here what you can see, it's a slice of a pavement cell. So it's a ZX, it's a cross section of one cells. And so if you already work with pavement cells at cotyledon, something you can show, it's the cells can be bumpy, can be curved. And so if I want to image the cortical microtubules, for example, I need to image all the surface of the membrane. And so even if we work just on cells and not in a whole a mouse brain, for example, we need a high number of slices because here in this image, I want all the cortical microtubules of these cells. I need to image 20 micrometer deep in the tissue. And so if I use this step of 0.5 micrometers, I need 50 slices. Of course, when we did imaging, we do imaging in the higher volume. We image all the cells, for example. And here, if I want to image all the cells, I image 50 micrometers. And so I produce 100 slices. And if I want to follow this cortical microtubule during time, maybe I will image more than just the cell because the cells can be moved up and down. And so if I choose the green dot lines, you can lose the cell during time. And so here in this example, when I did the time lapse, I image 60 micrometers, and so I produce 120 slices, which is not a little stack. And so it can be, and so here you can see the difference between the light sheet microscope, the alpha 3, and the other modality. So in the left, you can see the signal of microtubule for confocal with resonating scanning on. In the middle, classical confocal microscopy. In the right, the image for the alpha 3. So of course, it's a close-up in one cell in order to show you the signal. So the first things we can say is the time of acquisition is really competitive in the light sheet. With the resonating scanner on, it took 7 minutes to have these signals. With classical confocal imaging, with the optimized parameters, we take 1 hour and 19 minutes. And the alpha 3 took 1 minute and 21 seconds. So it's really, really faster. And so thing you can see is the signal in the alpha 3 in the light sheet microscope look like more the classical confocal microscopy images compared to the reasoning scanner. So it's really interesting if you want to do time-lapse imaging with high temporal resolution. And so if I come back to my image with my cells and I want to image with cells in a time lapse, I can do that with the light sheet with the alpha-3 microscope. And so something I did during my PhD is this. So it's to the goal is to follow the microtubules dynamics during time. And so here I will show you one videos of microtubules behavior. And it's a time lapse of 10 minutes and I took one image every 30 seconds. And so here you can see the dynamics of microtubules. You can see the polymerization of microtubules. So here you can see that. And so here it's not a really optimized imaging because I use a Z-tape of 0.5 micrometers. And so maybe when some experiments, you just need to use a 1 micrometer Z-tape, so the 1.5 micrometer Z-tape. And so if you use that, you can take one image every 10 seconds, for example. If you use some binning, you can do maybe one image every 5 seconds. So it's something I work on this optimization right now in the lab. And so we can do images, really fast images. And so, of course, I did some I quantified the dynamics. I did some analysis. And so I don't enter in the detail, it's just in order to show you we can analyze this image. And so something I can did is to do a heat map of dynamics of microtubules, where you can see in yellow the part where the microtubules are highly dynamics, and some part of the cell where the microtubules are more static. And so this microscope and this kind of microscopy, but the alpha-3 for the aerial organ of plants, you can do really fast imaging in order to follow fast biological process and do some quantification on this process. Just in order to show you other kind of labeling, we do some image of vesicles, and here it's a 3D imaging, so it's projection of the signal, but it's one stack, and so it's one image every second during 1 minute. And so we can see you can follow the vesicle during time. So it can be really interesting if you need to follow some vesicles. And so the temporal resolution is really high. And if you don't need 3D, but you really need really high temporal resolution, you can do imaging of just one slice during time. And here, for example, it's one image every 50 milliseconds during 1 minute. And so you can really follow the movement of the vesicles. And so thank you for your attention. I hope my talk was okay for you. It was interesting. I hope you you understand what you can do with the light sheet microscope and what it can be interesting with this kind of microscopy and the aerial organs of plants. So thank you.
Thank you, Matthew. I think we're going to try to take some questions now. I see a couple in there. Tim, I guess I'm going to drop back to you for the first question.
Okay, so first question I think that we got was, are there any references of X clarity and plant tissues? And the answer to that is not quite yet, but in about a couple of months, there is a different French group that will be releasing a paper published in one of the Nature publications. I'm not exactly sure which one, that deals with the X clarity and plant clearing and imaging.
Okay. question. There's a question that came in to the Q&A panel. Was that tissue still live for the growth pattern imaging? I think that was directed to Matthew.
Yeah, so thanks for the questions. Yes, the sample, the tissue is live for the growth pattern imaging. We can follow the growth pattern during time in the microscope.
Okay. Matthew, do you have any other questions that have come into you?
Yeah, so one question about roots. So the question is about roots. So I show a lot of image of aerial organs, and so the question is if it work for roots. And yes, of course, it work for roots. I didn't work on roots because it's not my PhD project, but I did some tests, and yes, of course, it's working for roots.
Okay. Tim, any other questions come in for you?
Nope.
No. Well, give it a couple of seconds here, see if there's a follow-up to Matthew's question. How much is the X clarity? Well, we don't usually talk about money on webinars.
So we don't, but if, yeah, exactly. And so if what you can do is we will, I think we have your registration information. So we will
Yep.
have one of the local reps get in touch with you, and they can talk about pricing and setting up a demo. And so that's the other thing that I forgot to mention in my talk is that, you know, we do free demos for the X Clarity system, you know, and so you will, if you are interested in one of those, simply write it in and we will get you to your local rep and they can get that arranged for you.
Great. Thank you, Tim. Well, I think that's going to do it. I don't see any other questions.
I think there's one more.
Okay. Yep. Go ahead.
I think this one is also from Matthew. Matthew,
Yeah. So the question is, would you explain a little about the illuminating lens for the light sheet? So it's two lens. It's one or two lens. So in the alpha 3, it's two lens, and this lens will produce the light sheets. And in the alpha 3, the numerical aperture is for this illumination lens, it's high, and so this induce a thin light sheet, which is interesting for the imaging of subcellular components, for example.
Thank you, Matthew. Well, I don't see any others. We can wrap this up. On behalf of Olympus, I'd like to thank all the attendees for joining us today, and Dr. Tim Stepp and Mathieu Cortes for their expertise and participation in today's event. We hope the material presented was informative and useful. Everyone who registered for this event will receive a follow-up email with links to the archived presentation, whether you attended or not. That's going to do it again today. Thanks for your interest in taking the time to attend this Olympus event, and we will see you again next time.